All right, hi, how you doing? Let's finish up coronavirus assembly. So, the point of this one is that we get to a point and we're not sure exactly what happens, but here's a nice diagram of the three reasonable explanations. And frankly, there are a lot of situations like this where we know that we have this starting point, this ending point, and the thing in the middle is not quite clear. And kudos to it's Paul Masters' group. He's just one of the best. They put together this figure which nicely illustrates all of it. So we know that the M protein is in the long conformation, and that's the M homodimer. Because that's the one that's associated with membrane bending. Now, it would never be associated with membrane bending this way. It's associated with membrane bending this way, but it's a diagram and whatever. They didn't get it all right. Next, we've got the main interaction partners. So we know that M interacts on various conditions, various experiments with both the packaging signal, the long bulging stem loop structure in the RNA, and with the small domain that they call N3 here, or it's the C-terminal domain. It's the last little bit, actually at the end of the C-terminal domain. So yeah, so that can either interact directly with M. The packaging signal can act directly with M, and then N3 can interact with M, or they can both interact together. They can all bind to each other. Who knows? Remember, there are several more or less identical binding sites, so you can potentially get four different N3M packaging signal combinations there together. And one way or another, getting those four together, we think like nucleates it. Like you put a little crystal in very salty water and it'll grow into a big crystal. Once you've nucleated binding of the genome to the N and M proteins, then the idea is that from that point it becomes much easier to bind the next one once you've got over the sort of energy barrier of binding the first one. So there you go. This is our special little way of making sure we bind the first, not even one, but four of those little molecular interactions that are going to effectively cement the genome to the membrane via M, and also via the nucleoprotein. So you've got discrimination because the virus is not picking up its own subgenomic RNAs. And you've got discrimination because the virus is not picking up negative strands, it's only picking up positive strands. So this is how you take a virus that makes both positive and negative strands, as they all do, and you pick up only the positive strand, and you end up with a positive stranded virus. Yeah, there you go. Because really, the distinction between positive strand and negative strand is one in the way that they start up, but two, it's the way that they end. Basically, which of the two strands they package. So yeah, stuff in the middle, fairly similar. All right, let's move on. So we can estimate the kinetics of this. We have good kinetics from HIV, which is like how fast all these parts happen. And so we can picture these little stages here from sort of the proteins are all at a membrane, but they're still flat. And then the proteins start to bulge, and then they bulge up, and then they're going to kind of sit down, and that's the point where we do scission and cut it off. And so based on the number of each of these stages that you can see in infected cells, you can tell roughly, like if you're looking at an assembly line and half the car parts are not assembled and the line moves at a pretty steady pace, then you can say, well, okay, we spend half our time over here and half of the time, after assembly, something like that. And if we know the entire process takes a certain amount of time, which is about 8 or 9 minutes for HIV, to arrive at the budding site, and then about 16 minutes until release, you can take that 16 minutes and divide it up based on which partially completed versions you're able to see lying around the shop. Yeah, it's kind of neat. So there you go. About 3 minutes for each one of these stages. The equations that govern this, you can describe anything with a good enough set of linked differential equations. That's not what this class is. If you want some of those, look down there in Schley et al. They've got all that stuff for you. But one model that describes this very nicely is derived from equations that are there to show what happens when you take a balloon and you press it between two panes of glass. Like, how does the balloon deform? And so, yeah, using that and using things that we know about the virus and how tightly the different proteins interact and how resistant they are to changes in osmotic pressure, for example, like the saltiness of the water around them, yeah, you can figure out a lot about the virus. And so what it ends up showing is that just like we saw with the actual virus, you'll start out like this. You'll get a sort of a growing crown of proteins that are, in this case, probably activated. So this is long M attached to the N protein. Maybe some of them have the packaging signal there, but there's probably only 4 out of the 100 or so M's that, actually no, several hundred M's that would have the packaging signal directly attached to them. The rest will have RNA bound up somewhere, maybe to the N protein. Then you're going to see it's going to rise up as we get close to the end, and then it sits right back down. And that's the point where we do scission, and that's a whole separate set of machinery that's going to be driven by the E protein, we think, and a whole different set of equations. The equations take you this far and no farther. Because it's a different set of proteins. You have to describe it differently. Yeah, one set doesn't do everything. So, there you go. So, rehashing this. So this is the current idea, and eventually this will be replaced by a better, more detailed idea. But until then, let's talk. The idea is that budding. You go from a situation with potentially a wider footprint in the inner leaflet of the membrane. By shrinking that footprint, so here we're taking these sort of flat amphipathic helices that are displacing a lot of membrane, they're sort of pushing it aside, and we're turning them so that they displace very little membrane. And so by doing that, we shrink the inner leaflet, and the outer leaflet stays the same size. And when you have those two things happening, the two leaflets stick to each other, and so they basically are forced to bulge. And as we do that and cross-link these together by basically having interactions with the nucleoprotein, then at that point you lock in the curvature. So membrane displacement to change the shape, and then protein interactions to lock that shape in. And so you've got these proteins that arrive, that just sit there for a while, and then boom, they start budding from the middle, and then they make a little thing that rises up, a little virion, and then sits back down, then scission, and then the virion can float away. So current understanding definitely will be improved on in a couple of years. Yeah, I don't know, check back, yeah, see where it goes. All right. I think that is all for... Oh, no, that's right. So this is just showing, once again, here's the M protein. So here's the short form. You see the short form is really wide. It's pushing these helices way out wider, and it's got an extra little hinge region that's kind of shoved up in the membrane. And the long form, we collapse this shape down to that shape. So it takes up less space on the inner leaflet, about the same space on the outer leaflet. And the hinge region that was all up in the membrane is moved to a different place. It's actually pulled down a little bit. The molecule's a lot taller, so it's hard to tell visually. You'd have to stack these two on top of each other. But yeah, that's the idea. All right, there we go. So the model is you have transmembranes that are going to hook our virus in there. You're going to have an anchor region which can be pulled out, like you can weigh anchor when you're ready to go. And so that's going to be our hinge region here, as well as the helices that start wide and then come in, snap in sort of narrow in the long conformation. And then you have the activator, which is the C-terminal region. It's all these beta strands down here that make these little sort of beta baskets, if you like. Those are the thing that's going to respond to the presence of the N3 domain and the viral RNA. All right, there you have it. Coronavirus budding in a nutshell, as far as we can say. All right, that's good enough. Thanks very much.